Ok, allora, se vuoi farti vedere, you have to stay.
Ok, faccio vedere.
So, I will put it like this. Okay. So welcome everybody to this new series of neuroscience seminars. And today we have Pierre Magistretti, who is an international recognized neuroscientist who made a significant contribution in the field of brain energy metabolism by discovering several molecular and cellular mechanisms that underline the coupling between neuronal activity and the energy consumption in the brain, and by revealing for the first time the key role that astrocytes play in this physiological process. He has an impressive record of publication, and his work is particularly relevant for understanding the origin of the signal detected with the brain imaging technologies, not only for this, but also to let's say, for revealing the role of the associated metabolism in neuronal plasticity and neuroprotection, as we will hear today. He received many recognition during his career. For example, in these days, he's here because he has been elected foreign member of the Accademia Nazionale dei Lincei. He was president of the France of the IBRO. In Switzerland, he was a director of many neuroscience departments, including the Brain and Mind Institute at the TFL in Lausanne. Then he moved in Saudi Arabia at the KAUST University, and he was a dean until 2 years ago. And in these days, he is a director of the Smart Health Initiative. Okay, so today we present a Neuron-glia metabolic coupling mediated by lactate, and we will hear a lot about this pathway in plasticity, in memory, and in neuroprotection. So the floor is yours. If you need the microphone there.
Yeah. Okay. Well, thank you very much, Paola. I am really very happy to be here today for many reasons. The first one is, I think it's my first lecture live in maybe 18 months. I've given several, but all by Zoom. And so it's really nice to see real people here. So that's the first. And so I thank Paola and the members here who invited me. The second reason is more personal. I was telling Tonino that actually I spent a few years, I was born in Milano, but left Milano when I was 10 and went to Switzerland. I stayed there, went in the US, came back. But of course, I always kept strong links with Italy. And I was here in Rome. I studied in Rome, 2 anni di ginnasio e 1 di liceo, al Liceo Mameli, so in Parioli. And my mother was a young professor here. I don't know what the title was at the time, but let's say assistant professor here at the University of La Sapienza in the Institute of General Physiology, next door. And so I would come, take the bus, the tramway, and come here. And so it's very personal for me to be here. And and finally, as Paola mentioned, I'm here for a very rewarding event this afternoon and tomorrow in the Accademia dei Lincei. So all this being said, let me tell you our story that really started over 40 years ago when I was a graduate student at the Salk Institute, working with, probably you don't know him, but more senior people here know him, Floyd Bloom. He was really one of the main neuropharmacologists at the time, and neuroscientists in general. He brought together many disciplines. His lab was very large. It was like, however, like a small neuroscience department. And that has been really a fantastic experience. So I've been interested since then in the mechanism at the cellular and molecular level of brain energy metabolism. And as you know, the brain uses a lot of energy, 20% at least of the body's energy goes to the brain. There is also this local regulation of energy, which has allowed the development of functional brain imaging, because what you measure with PET scans or fMRI is actually the use of energy through increases in blood flow, glucose utilization, not directly electrical activity. So it's an important topic. And in fact, it was, let me see if I can move it. How do we move this? Okay, good. So in fact, this notion of local regulation of energy delivery to the brain was formulated already at the end of the 19th century by one of the founding fathers of modern neuroscience, Charles Sherrington. And he postulated that the brain, let me just move this if I can, so I can see. Yeah. Can you move this? If possible. Se si può muovere questo qua. Okay, okay, that's better, that's better, that's better. Okay. So he formulated this notion that the brain, that's in yellow, the brain possesses an intrinsic mechanism by which its vascular supply can be varied locally in correspondence with local variation of functional activity. This was really very clear formulation, in this case for vascular supply, but as we also now know for many years, since the 70s, the work of Louis Sokolov, he used 2-deoxyglucose as a marker for glucose utilization. And there is also this coupling between activity and glucose utilization. So the question that I have been addressing over the years was actually the one of what are the mechanisms? No, it's okay, it's okay. Okay. What are the mechanisms that allow this coupling? How to relate the electrochemical events that occur at the synapse? Yeah, good. With the metabolic and vascular responses, and then produce the signals for functional brain imaging. And the classical view had been that somehow neuronal activity would result in the release of some kind of vasodilator, act directly on capillaries, which would then increase their blood flow there, and that energy substrate would come to the neurons, the active neurons. However, this view was missing an important player, which we discovered many years ago, and many now have confirmed these observations, that astrocytes are key in this coupling. In fact, you may know astrocytes are part of the general class of glial cells, which comprises also oligodendrocytes that make myelin, and also, among other things, and microglia, very important for immune and inflammatory responses, but also in synaptic pruning. So now there is a vision that the neurons are very important, of course, but the other half of the brain, as I like to call it, is just as important. I say other half because depending on the quantifications, there are more or less as many neurons as glial cells. So how did we get to this observation? Well, first, just some consideration. If you take the astrocytes, you can see that they have features that are very, very useful, let's say, to couple synaptic activity with energy delivery. Because first of all, they have processes. You have here a presynaptic profile, a postsynaptic one, and here in blue, these astrocytic processes around the synapses. In fact, in the rodent, one astrocyte can have this arrangement around synapses, around over 100 synapses, even more. So clearly they are positioned and be able to sense synaptic activity, not only because they are at the right place, but because they also express reuptake sites for transmitters, in particular glutamate, but also receptors. And this ensheathment by astrocytes around synapses is dynamic. It's not there once and forever. It's plastic. There can be more or less. And there are a lot of studies, in particular with Graham Knott in Lausanne and many others, showing that activity modulates this coverage of synaptic profiles by astrocytic processes. And then the other feature, the astrocytes have processes around capillaries. They're called the end feet. And as you can see here, essentially, the entirety of capillaries are covered by this end feet. And here you have an EM image here with a very nice end feet that is abutting on the capillary. This is work by Corrado Cali, very, very good neuroscientist, was with me for many, actually with Paola, he got his PhD with Paola, and then worked with me, and now is assistant professor in Torino. So this is the more artistic view. Again, you see these processes around synapses that allow astrocyte to sense synaptic activity, and these end feet that cover the capillaries. So in our work, we described the fact that actually astrocytes, and we showed the mechanism, molecular mechanisms that underpin this coupling. And I will give you a few examples of the relevance, sorry, of this arrangement, of this function for neuroenergetics, brain energy metabolism, imaging, plasticity, and some ideas about disease. So this is me. I like to show this picture because it shows, first of all, my mentor, a man I greatly admire. Unfortunately, he's not in great shape at the moment, Floyd Bloom, and myself when I was 28 or 29 at the Salk. As you can see, many things have changed. You could be in a lab in shorts and no white coat. And yeah, well, that's not the only thing that changes, you can see, but that's fine. I mean, I spent really wonderful almost 4 years there. And so we described in 1981, the fact that some neurotransmitters, VIP, which had just been discovered in the brain, like many peptides. That was the time, late 70s, where suddenly from 2 or 3 neurotransmitter, 4 or 5, you know, GABA, glutamate, the monoamines, noradrenaline, dopamine, serotonin, acetylcholine, suddenly there were 50 more transmitters, and these were peptides. So the lab of Floyd was trying to understand the role of this peptide. So I ended up working with VIP for many reasons, which I like to tell, but now it's not, we don't have time. But anyway, we showed that VIP and noradrenaline were promoting glycogenolysis in the cortex, which was kind of a strange idea why we did this. And again, I won't go in details, but I can tell you that it was very bit by chance and also some ignorance, because if I had known that there was very little glycogen in the brain and it was all in glial cells, I probably would not have done this. So I didn't know that. And for many reasons, I decided to try whether VIP was glycogenolytic, mainly because it was glycogenolytic in the liver. So I said, maybe there is an analogy there. But that was one of the first demonstrations that a molecule released by neurons, a neurotransmitter, was acting on glial cells. That was the first novelty, if you wish. And the second one was that this effect, not surprisingly, was not electrophysiological excitation or inhibition, obviously, because glial cells are not excitable, but it was like a metabolic and endocrine almost effect of these neuronal molecules on glial cells. And there was even a physiological meaning for this, delivery of extra energy. So we found actually that this was mediated by very few transmitters, either an analog of VIP, PACAP, or a few, noradrenaline, or some purines, adenosine and ATP. And essentially, all this was mediated by second messenger cyclic AMP, like it was done happening in liver and muscle. So from this, we concluded that the brain possessed a system that would allow neurons to send a signal to astrocytes, mobilize some energy reserves in the form of glycogen, and promote glycolysis and provide extra energy to neurons. And because we were also studying the morphology of the neurons that contain VIP, noradrenaline was known at the time. I'll tell you in a moment how it looks for those who don't know. But VIP neurons, we described with John Morrison that these were bipolar cells. So you can see them here in green. These cells actually define what one would call cortical columns, receiving inputs from other cortical areas. So we proposed that this was a system to release, to mobilize energy within cortical column to match the increase in activity in these columns. But also the noradrenergic system, in contrast, which, as some of you may know, the cell bodies are outside the cortex in the locus ceruleus, and they project diffusely across the brain, like a sprinkler, if you wish. And so we proposed that, in contrast to the VIP neurons, there were this noradrenergic system which would kind of prime the cortex globally to activity. And in fact, the locus ceruleus is particularly active during arousal, during attentional events. So it made sense that something global like this would be needing energy. So a few years later, when I came back to Switzerland, I set up my lab. I had a few students and postdocs, and with Luc Pellerin, who came from Montreal, from the Montreal Institute of Neurology, we described another mechanism that couples activity to energy, acting on astrocytes. And this was by glutamate, which, as you know, is actually the main excitatory transmitter in the brain. About 70 to 80% of the synapses in the cortex use glutamate. So whenever there is an activation of a modality, glutamate will be released. And we showed that the uptake of glutamate into astrocyte, and this was already known. It was known that glutamate, once released by neurons, is very efficiently removed from the synapse into astrocyte. And this, of course, serves two purposes. One, to maintain a good signal-to-noise ratio. So whenever glutamate comes into the synapse, there is no more glutamate, and so it's a signal. And also excessive glutamate, as you know, can be toxic, excitotoxicity. So that's a very efficient system that astrocytes have in place to modulate transmission and to protect neurons. But what we showed that not only this happened, but this uptake of glutamate into astrocytes would promote aerobic glycolysis. So what's aerobic glycolysis? That's actually the production of lactate in the presence of oxygen. It's known also as the Warburg effect. It's typical of many cell types, particularly cancer cells, but also other cells, immune cells. So it's this lactate can be formed not only because of hypoxia or ischemia, so anaerobic glycolysis, but actually through a physiological mechanism. And by the way, this engagement of lactate, description of the effects of lactate, was received initially with a lot of criticism because, you know, lactate is bad. But actually, it's not true because many, even Henry McIlwain, we don't know him, but he was one of the early neurochemists in the 50s. He showed that lactate is just as good as glucose to maintain activity of neurons in vitro, in slices. So anyway, so this is the mechanism that we described over here. Again, the synapse, the astrocytes, and the capillary. So what we showed is that this release of glutamate and its uptake into astrocyte, which, by the way, is driven by the electrochemical gradient of sodium. So one glutamate comes in with 3 sodium ions. Then glutamate is actually recycled, at least in part, back to neurons through the glutamate-glutamine cycle. Glutamine is inactive electrophysiologically, but then once in neurons, it can be converted to glutamate and then replenish the glutamate pool. But this costs energy. Transforming glutamate to glutamine through glutamine synthase costs 1 ATP per glutamate. Then sodium, of course, 3 sodium ions come in. That's like in any cell, the sodium-potassium ATPase is activated. And that also will cost energy, 1 ATP to extrude 3 sodium ions. So whenever one molecule of glutamate is taken up by astrocytes, that will cost 2 ATPs to to the astrocyte. And that will do like anything in any cell. It disinhibits enzymes such as hexokinase or phosphofructokinase, and then glucose is sunk into the astrocyte or in any cell where there is this this process, this glycolytic process. And what we observe is that lactate is produced, and is produced, just keep in mind, astrocytes have mitochondria, so they could oxidize lactate, transform it to pyruvate and use it. They use some, but most of lactate, glucose that is taken up is transformed to glutamate, to lactate, sorry. This doesn't mean that there is no glucose uptake into neurons. There is GLUT3, the transporter GLUT3, astrocytes have glucose transporter 1, but I'll show you what is the role of this glucose that goes into neurons. But during activation, when glutamatergic activity is going on, then there is definitely a use of uptake of glucose into astrocyte and production of lactate. So actually, we showed with Jean-Yves Chaton very briefly that indeed the sodium-potassium ATPase is very important. We could see in blue ATP consumption increases and here decreases. In red, sodium levels. So when you present glutamate to astrocytes, both go up, sodium and ATP utilization. If you block the pump, immediately ATP consumption decreases and sodium increases. So these mechanisms are really very tightly coupled. And so this gave rise to what is now known. If you don't mind, I'll take off my jacket, it's a bit warm. Gave rise to what is now known as the astrocyte neuron lactate shuttle, whereby a neuronal signal, glutamate, actually is detected by astrocytes, and through the process that I just described, and we described many more. In particular, we described that lactate is released through monocarboxylate transporter 1 and 4, mainly 4, and neurons take up lactate through monocarboxylate transporter 2. Then also the lactate dehydrogenase that transforms pyruvate to lactate is only present in astrocytes, and the LDH1 is present, and neurons contain only LDH1, which favors the transformation of lactate to pyruvate. And then pyruvate can be used by neurons through oxidative phosphorylation. And now this ANLS, as it is called, as more actors. I mean, there are other signals that have been shown, mainly by the lab of Felipe Barros in Chile, showing that other signals that are increased during activity, potassium and ammonium, can trigger the ANLS. And they also describe the high-capacity lactate channel in addition to the transporters that will allow lactate to be released. And there are really, obviously, molecular basis for having astrocyte being mostly glycolytic and neurons mostly oxidative. Well, first of all, I mentioned it in passing, astrocytes contain glycogen, neurons don't. Then astrocytes have a high expression of this enzyme, phosphofructokinase fructose bisphosphatase 3, PFKFB3, which really is a turbocharger for glycolysis. Neurons have it, express it, but very transiently, it's permanently degraded by the proteasome. So, and if this is blocked, if PFKFB3 activity in neurons is kept high, actually neurons die. It's toxic to neurons. So neurons cannot afford to have a high glycolytic activity, and I will tell you why in a moment. Then astrocytes express protein kinase, the M2 form, which is typical of glycolytic tissues, whereas neurons express PKM1, which is typical of oxidative tissues. And at the end, also pyruvate dehydrogenase that allows pyruvate to enter the Krebs cycle. This activity is low in astrocytes, and there are many reasons for this, which we can discuss, whereas in neurons, it's high. So it favors the use of pyruvate in the Krebs cycle. And essentially, this is work by Salvador Moncada and Bolanos and their group. In a nutshell, glucose in neurons is used activate as a substrate of the pentose phosphate shunt. If you remember your biochemistry, glucose can enter either storage in the form of glycogen, or go through the pentose phosphate pathway, or go through glycolysis, and then eventually the TCA cycle is spiraling. So neurons use glucose mainly for the pentose phosphate shunt. And what does the pentose phosphate shunt? It produces NADPH, which is a reducing agent, which allows the regeneration of reduced glutathione, which is the key molecule for scavenging reactive oxygen species. And neurons are very oxidative, so they produce a lot of ROS. So that's the conclusion that, yeah, astrocytes are mainly glycolytic and neurons mainly oxidative. Work by Felipe Barros, Bruno Weber, and we also collaborated in this, but this is really mainly their work, showed that there is actually spontaneously a lactate gradient in between astrocytes and neurons. So I will not go into many details, but when this is in vivo imaging, and they can monitor lactate and with a particular genetically encoded fluorescent dye marker, laconic it's called. So you can have astrocytes that express laconic and detect lactate, or neurons that express laconic and detect lactate. And what you see when you open the transporters, MCT1 and 4 in astrocytes and MCT2 in neurons, so now you have a free flux of lactate. Well, what happens is that there is an initial decrease of lactate into astrocytes and a delayed increase of lactate into neurons, showing that when everything can flow freely, there is already a natural gradient of lactate between astrocytes and neurons, plus this becomes even more important whenever there is more lactate formed in astrocytes. So let me now go to the next step, having given you a background on neuroenergetics. So what is the relevance of this for functional brain imaging? So we undertook a number of studies where we said, well, if this glutamate uptake into astrocyte is so important to allow glucose uptake into astrocytes, well, if we were to block or downregulate the expression of this glutamate transporter and activate neurons, then we should have also a decrease in this glucose uptake, because the signal would be impaired. And that's what we did. We used a very classic pathway, the whisker to barrel pathway. It's a sensory pathway originating here on the face of the mouse. And for each vibrissa, little vibrissa, there is actually a very specific projection to the somatosensory cortex of the mouse, called the barrel field. That's in layer 4. There are neurons arranged in a certain manner that can be detected. And then there is a one-on-one actually correspondence. In fact, the former chairman of the department where you were many, many years ago in the 70s and 80s, Hendrik van der Loos, described this whisker to barrel pathway. And so you can actually very elegantly stimulate one vibrissa and then do 2-deoxyglucose autoradiography and see if the corresponding vibrissa is using more glucose or not, because you have activated this pathway. And so we use this, and by the way, we collaborated with Egbert Welker, who was one of the students of van der Loos. What you see, you can just look here at the bottom panel. On one side, we had injected oligonucleotide antisense that downregulate the expression of the glutamate transporter, which is essentially only in astrocytes, stimulated the vibrissa, and looked how much glucose we get to the corresponding barrel field. And as you can see here, in the control experiment or control side, you see a nice imaging actually of 2-deoxyglucose, like PET. This is autoradiography. And but very little on the side where the glutamate transporter on astrocyte was downregulated. So this is an in vivo demonstration of this coupling. And then there is another one very quickly. We used also some knockout mice for these transporters, the glutamate transporters, and they actually have much less uptake of glucose on the in the cortex. And these mice, by the way, they have two problems. They are epileptic, and which is not surprising because if you cannot remove glutamate, you have hyperexcitability. And two, they actually have a massive neurodegeneration very early on, showing again that glutamate is toxic, which was known. So then finally, in the lab of Yori Buzaki, he used or actually Julien Choquet used a dye, a glucose, a fluorescent form of glucose, and then looked at stimulation in vivo with cellular resolution, with confocal microscopy, and would see that under basal conditions, essentially there is mostly of glucose is in the blood, in the capillaries. There is in astrocytes and neurons, a little bit more in astrocyte, but not significantly. But during stimulation, actually, the fluorescence glucose is much more accumulated in astrocyte rather than neurons. So all these are evidences to support the idea that whenever there is an activation, glucose will mainly enter into astrocytes because of this neuronal signal. And so when one looks at the fluorodeoxyglucose PET scan, what one is seeing during activation, certainly, is uptake of glucose into astrocyte. It doesn't remove anything, any validity to the method for localization because the initial signal is the glutamatergic activation. But it's interesting to consider that whenever there are abnormal PET signals, one should consider that maybe, most likely actually, there are some deficiency in this transduction mechanism. And a couple of years ago, Luc Pellerin and people at Montreal Neurological Institute showed that in vivo, using PET now, what they did the opposite of what we did. We downregulated glutamate transporter. They upregulated pharmacologically, one can do this pharmacologically, and they see an increased PET signal. So if you increase the amount of glutamate transporters on astrocyte, you will take up more glucose. So let me now move to the more recent work, although this also in part was recent, the role of this coupling in neuronal plasticity. And so we reasoned in the following manner. We said, well, memory and learning is based on neuronal plasticity. A lot has been studied in this process, increased release, increased receptors, induction of genes. But what we asked was, is this coupling, metabolic coupling between astrocytes and neurons also plastic? You know, whenever there is plasticity in neurons, will there be an adaptation in astrocytes? Or even more provocatively, is the astrocytic plasticity necessary for neuronal plasticity? And so we devised a few behavioral experiments, either the spatial learning or the inhibitory avoidance behaviors. I'll tell you more about the latter in a moment. But essentially what we did was detect glucose uptake, which is a marker of activity, in a way, the marker of the astrocyte neuron lactate shuttle, before and after learning. And this mapping allowed us to see areas that are involved in learning because there is a differential between before and after. And then we could micro dissect these areas and look by simply RT-PCR whether the genes involved in the astrocyte neuron lactate shuttles are modulated, their expression is modulated by learning. And indeed, we found this was work by graduate student Monica Tadi, and we showed that several genes involved in the astrocyte neuron lactate shuttle, so in Neuron-glia metabolism, their expression is upregulated by learning. And a very interesting, at least for me, observation was that essentially all genes involved in glycogen metabolism are upregulated by learning. So that brought me, this was in the early 2000s, so maybe 15 years ago. It brought me back to the 80s when I was in short in the lab, because that was glycogen, I was working on it. And so we decided with Cristina Alberini, who at the time was at Mount Sinai, but now at NYU, an expert in molecular mechanisms of learning, to block glycogenolysis, in other words, glycogen mobilization, or block the transfer of lactate from astrocytes to neurons during learning and see whether this affected learning. And this was a paper that got a lot of visibility because we actually showed that if one blocks the transfer of lactate from astrocytes to neurons, one blocks memory consolidation. And why was it quite, got a lot of traction? It's because, again, in a neurocentric, as we were saying with Antonino, world, suddenly bringing into the picture that astrocytes, glue, inert cells, and lactate, metabolic end product, were important for memory was really a shock. But we and others, and that was confirmed by others. And so that's an interesting observation. Let's put it like this. And so very briefly, what we did was with Cristina, we used this inhibitory avoidance test, which is one trial learning. So 2 boxes, one with light, one without light. And as you know, rodents like to go in the dark. So when you put them in the light, very quickly they move into the dark. The day of training, when they go into the dark, you give a little electric shock. Next day you test them, and they take much longer to go in the dark because they kind of remember that something not so pleasant happens. So what we did, we essentially either blocked glycogenolysis with this molecule, the AAB, or we downregulated the expression of MCT1 and 4, the astrocytic ones, and MCT2, the neuronal one. And just, I will not present all the data because this has been published a while ago, but look just as these 2 ovals. Here is the control condition. So the animals have a long latency before they move into the dark. And when they receive DAB during training, so they cannot mobilize glycogen, they cannot use glucose, lactate, sorry, they have a very short latency. So it means they don't remember. And here, when you, at the same time as you give DAB in the hippocampus bilaterally, you also give lactate, there is a complete, almost complete rescue. So by bypassing, if you wish, the production of lactate by astrocytes, by just providing lactate here, you rescue consolidation. And I don't show the data, but just for you to know, if we downregulate 1 and 4, so we prevent the transfer of lactate from astrocytes to neurons, we also block consolidation, and it's rescued when we give lactate. But when we downregulate MCT2, we also block consolidation, but we cannot rescue with lactate because obviously lactate cannot enter the neurons. So just for you to know, during learning, I'm going to be very quick, by microdialysis we saw, and this is interesting, it lasts for quite a while. There is a massive increase in lactate, well, a significant increase in lactate in the hippocampus during learning, and this is completely blocked by DAB, meaning that the lactate comes from the astrocytes. But also LTP can still be induced, in vivo, LTP can still be induced by the normal activity, that triggers LTP, but the sustainability of LTP decreases immediately if lactate is not formed and it's rescued by lactate. So in summary, what we show here is that lactate is important for memory consolidation. It's formed during learning and it's important for memory consolidation. So with Cristina, we went on. This is really work that was done mostly in her lab with Gao, a nice student who was at EPFL and then went to, very competent, very really amazing student, Virginia Gao. And she showed that actually if you block consolidation with propranolol, which is a well-known phenomenon, so you learn, you do a training learning, and then you block after learning with propranolol, you don't have consolidation. And this is because noradrenaline is considered to be important for memory consolidation. There are many data showing this. So we reason, wait a minute, noradrenaline is glycogenolytic, as I showed you at the beginning. So it means that whenever noradrenaline is activated, there is glycogen mobilization and lactate production. So how could we reverse the blockade by propranolol, which blocks beta-adrenergic receptors, by providing lactate? And the answer is yes. So this is, again, the latency control. This is in the presence of propranolol, essentially no consolidation. Then in the propranolol plus lactate, we reestablish. And then here, well, this is glucose. You can reverse partly with glucose, but with high doses, and it's much more transient. And by the way, also, the previous work that I just showed, we could rescue with lactate, as I showed you, but essentially we could not rescue with glucose. So the idea is that actually noradrenergic systems through glycogenolysis acting on astrocytes actually is important for memory. I mean, that's probably the mechanism for its memory consolidation. So we very briefly, we wanted to see if this role of lactate in memory was only for aversive memory. So we looked at condition place preference to cocaine by using, as you know, you inject cocaine in a side of a cage, which is very unique with the special, I mean, a very marked environment, and then you inject it vehicle in another compartment, and so they have a choice. And then they go where they where they took cocaine. And again, you see here, you have a clear preference in the control animals, completely blocked by DAB. They have no preference, they just go either side. And then now with lactate, that you recover. This is actually, these are the three colleagues, Benjamin Boutrel, Anthony Carrard, and Benjamin Bourjabot, and I was not there the day the picture, so they put my picture in the back. That was kind of that. So now the question is, okay, glycogen seems to be very important for this memory consolidation. So we were wondering, where is glycogen localized in the astrocyte? Is it diffusely present, or is it closer to the synapses, in the processes around the synapses, or maybe in the and feet, you know, where actually glucose comes in. So this is really nice work done by a nice, really great postdoc, Corrado Cali. I told you already that he's now in Torino as an assistant professor. So he was trained in Lausanne with Paola, but then worked a lot also with Graham Knott, who's a specialist of EM and 3D reconstruction. So he performed the 3D reconstruction, and with many master students and interns did what is very tedious work, which is segmentation. So at each section, drawing what is an axon, what is an astrocyte process, what is a, and they also even now are developing semi-automated AI-driven machine learning processes, but still a lot of supervision is needed. But anyway, so they were able to do this reconstruction, and you can see in green here all the surfaces and volumes occupied by the astrocyte, which is quite impressive. But even more, and this is really something that was developed, I hope it works at KAUST in what is called the cave. You can use this 3D data, this segmented data, and transform this into a 3D virtual reality environment. I don't know how it's done. I'm not very good at computation, but anyway, Corrado is very good. Before Lausanne, he graduated from Politecnico di Torino. And so you can really, let's see, we can make it work. Yeah, so you have now this block of hippocampus, and you can walk into it. So you see first all the elements, that's easy. You see in green again, how many, you can even, how many astrocytes there are. In purple, you see the glycogen granules in the astrocytes. And here is Corrado in the cave, walking in this virtual reality. So he's in the capillary now. And then you see he's moving behind the capillary, and at the bottom you can see this is real data. You see an EM section. So he's moving from the astrocyte behind the astrocyte, and now he's going to enter into the astrocyte. Here you go, and he can see by transparency, because you can do what you want with this data. He can see the dendritic spines, and he can see where the glycogen granules are in respect to the spines. And of course, it's nice and kind of spectacular, but it's also very useful, because essentially he could see that by this walk that there were clusters of glycogen granules. And when he did the quantification, and by the way, we published this in a classic neurohistology journal, J. Comparative Neurology, and actually made even the cover, because it's really, I think, a very innovative way to do EM and quantitative EM and neurohistology. And now it's becoming simpler. You don't need a cave. You can do it also with the Oculus type goggles and things like that. But anyway, so what you see is that essentially 2/3 of the glycogen granules were either, I mean, in astrocyte, but close to spines or close to boutons, most likely monoaminergic boutons where noradrenaline is released. And more recently, Corrado worked with colleagues in Milano and did a study that possibly had been done before, but we actually couldn't see any real solid data on this. And then to look at learning paradigm, novel object recognition, and then look at the density and size of dendritic spines and glycogen granules in the astrocyte near the dendritic spine. And what you saw is that this is in terms of the number of spines per cubic millimeter in control. After learning, it increases. But if you block with DAB, you block lactate production. Actually, you don't have this increase in spines, and this is reversed by lactate. You have also, by the way, an increase in the size of the spines, but actually it turns out that lactate does not have an effect of this. And again, he also looked on the other side at the glycogen granules, and there is also here an increase in glycogen granules following learning. And this is quite interesting, I think, and we can debate why. And this doesn't happen with DAB, which is not surprising. So in summary, what you see that you are in control, you have dendrites with a certain number of spines and some glycogen granules in the astrocytes. Learning will increase the number of spines and their size, and the number and size of glycogen granules near these spines in the astrocytes. DAB blocks this, and lactate rescues only the number of spines. It doesn't rescue the glycogen granules because it's not surprising, there is no gluconeogenesis in astrocytes. So it's still work in progress, but it clearly shows that there is a metabolic reprogramming in astrocytes following learning, and maybe that's necessary to sustain the number and size of spines following learning. So let me get to the final part of my talk. And so based on the work with Christina and Suzuki as a first author, a postdoc, very nice postdoc from Japan, Hideaki Suzuki, now he is back there for a while. As I said, with glucose, we could not rescue, as we did with lactate when we blocked the transfer of lactate from astrocyte to neurons. And so we wondered, well, maybe there is something special with lactate. It's not just energy. because, you know, I wouldn't say the criticism, but people would say, yeah, well, big deal what you find, because after all, if a car, you remove the fuel, it doesn't move. So if you remove energy, it will not move, which I thought was very nice because it was implicitly acknowledging that lactate was a energy substrate for neurons, which again, in the 90s, it was not at all accepted. But anyway, what we found, we went back in vitro and we looked at on astrocyte what lactate or delactate or pyruvate or glucose were doing. It turns out that, and we looked at genes that we knew are involved in plasticity. So what we saw is that ARC, ZIF, also CFOS, but this is more an activity marker, but ARC and ZIF, and also BDNF later, about 2-3 hours later, this is within, the ARC and ZIF is within 1 hour, 30 minutes, 1 hour, were induced by lactate, by L-lactate, but not D-lactate or pyruvate or glucose. So there was something definitely special, and this was occurring at concentration between 2 and 5, even 10, even more of lactate, which is easily found in the brain. And after a long search, we said, how is it working? And actually, this is work for a postdoc who was there, and he's a very, very old postdoc. He's been 20 years working with me, but a very, very essential element in the lab, Igor Alamo. He and others in the lab showed that actually this effect of lactate on gene expression for plasticity is mediated by facilitating or potentiating NMDA receptor signaling. So, which makes sense in a way, because NMDA is really the quintessential, one of the important elements for plasticity. And we have since, I mean, this is about 5-6 years ago. And so here you can see that the current, the NMDA current, this is the control, this is in the presence of lactate, this is calcium. So it's really like a turbocharger of the NMDA receptor. It activates the cascade of signal transduction involving ERK1/2, so as expected. So there is more, but it's been published, so I will not go into details. But essentially, the message is that lactate is not only an energy substrate, but is also a signal that works through an NMDA receptor potentiation, maybe other mechanisms. And we are working very hard, and it's going very slowly, but we're trying to understand the molecular basis of this. And what we can say already is that it's the NR2B subunit that is involved in this potentiation. And essentially, and just this is a bit early, but you know, we have more and more data converging on this. It facilitates the interaction between NR2B and kinase 2. So essentially, that's how lactate works. And this is mediated. I mean, it involves the reducing capacity of lactate, because when lactate is transformed from lactate to pyruvate, there is NADH that is formed. And so the redox state of the environment changes. So this is how we are thinking. Actually, more recently, Michael Margineanu, very nice postdoc from Romania at KAUST, and Hubert Fiumelli, also someone who's been with me for many, many years, they showed that using an unbiased approach, transcriptomics, and with a lot of bioinformatics behind, because you get a lot of false positive or negative with transcriptomic. At least we thought we needed really many different algorithms to come up with a list, which I am quite confident that you know, I would bet some money that it's correct. And we saw that some genes involving synaptic plasticity and neuroprotection are induced by lactate in neurons, such as ARC, BDNF, I already showed you. Interesting, NPAS4, also VEGF and PAS4, and some are downregulated, such BCL2, and very importantly, we're very much interested at the moment in TXNP, which is involved in redox regulation. And we are also, this is work by Hubert Fiumelli and others in the lab. They essentially, what we see is that this is again a patch clamp for the glutamate current. In the presence of lactate, the peak is larger, and also the repolarization takes longer, the tau is longer. Interesting, pyruvate also increases the tau, but no effects on the peak. So we are working around all these mechanisms, and I'll tell you a bit more. So let me go to the final part, what's the relevance of all this for diseases. For many reasons that are outlined here, we became interested in see whether lactate would be involved in a disease which has some grounding in plasticity, and that's depression. There is inhibition of neurogenesis, there is also cognitive impairments, and there was also a set of literature data showing that, first of all, there is a decrease in density in glial cells in the dorsolateral prefrontal cortex, subgenual in depressed patients, that impairing astrocytic viability with toxins in rodents also results in depressive behaviors. We had made this observation which we published in a small paper that some, not all, but some fluoxetine and paroxetine antidepressants, not the tricyclics, had a metabolic effect on astrocytes. They actually were stimulating lactate production and glycogenolysis, and epidemiological data showing that exercise is actually it's an improvement therapy for depression, and exercise, of course, will result in lactate production. And so we published, we did this work. It was work by Anthony Carrard and Jean-Luc Martin. He's also someone who stayed with me still now. We're working together. This is us in 1986, and we still work together. So it's another, story how science can bring people together for many years. And what we showed, is that peripheral administration of L-lactate, IP, produces antidepressant-like effect in different models of depression in rodents. So the acute force swim test, it's kind of a strange test. You put the mouse in some water, and after a while they get very passive. So it's not very subtle, but on the other hand, it's a very good screening for antidepressants. All antidepressants prolong the activity. Another one, which is a little bit more physiological, is the open field force swim test, where you put them in a pool, and after 5-6 days, you build up this passivity, and then you give them antidepressants, and within 10 days, 2 weeks, they actually become active again. And lactate works exactly as dizipramine. And then finally, chronic corticosterone, which is a model of stress-induced depression. Actually, stress and high corticosterone levels, at least in the subpopulation of depressed patients, is a risk factor for depression. And so you can generate some depressive behavior in rodents by chronic injection of corticosterone. And so we found that lactate in all 3 models has an antidepressant effect, exactly like dizipramine. And also we looked at the molecular expression of genes, and we found that there are a number of genes whose expression is modulated by lactate. It's changed by these treatments and kind of reestablished with lactate. And more recently, I just jumped through the data because it's almost to the end. Very recently, we published again in Molecular Psychiatry this paper showing that actually for the antidepressant effect of lactate, neurogenesis is required. In other words, if we block neurogenesis, well, first of all, this is the number of surviving precursors. It's decreased by chronic corticosterone. This is well known, but this is reestablished in the presence of lactate. And if we use this molecule, temozolomide, which blocks neurogenesis, essentially we lose the antidepressant effect of lactate. And we are again thinking that NADH is important because none of these antidepressant effects of lactate is mediated by pyruvate, is mimicked by pyruvate. And the only difference between lactate and pyruvate is the formation of NADH. And we saw that beta-hydroxybutyrate, which by the way has been described as having antidepressant effect, and we validated this, when it's transformed to acetoacetate, also forms NADH. So we think really that lactate and NADH signaling are important in the effects of lactate. And just think of this, it would be a very smart way of nature. The same molecule, by being transformed to pyruvate, produces NADH, which is, of course, participates in the oxidative phosphorylation and produces ATP, so it's energy. But at the same time, this reducing equivalent will modify the redox state of proteins and play a role as signaling molecule. So I think I will just mention this. We saw that in ALS models, this is work we did with Jeff Rothstein, there is a decrease in monocarboxylate 1 in spinal cord in these SOD mice. And if we block MCT1, there is axonal damage, so some relevance for other aspects of disease. And Klaus Nave in Göttingen did a lot of work on the role of lactate released by oligodendrocytes. And one can imagine that lactate can be formed by astrocytes. And as you know, that oligodendrocytes are coupled to astrocytes through gap junctions. And so this lactate that oligodendrocytes release on axons and is necessary for the axonal integrity. This is work by Klaus and his group. Again, highlights the importance of lactate through another mechanism. And then let me just conclude with this. So I think I've showed you that there is a very active dialogue between astrocytes and neuron, metabolic dialogue. Astrocytes respond to neuronal signals and they send back many signals most likely, but certainly lactate, which has a dual function as an energy substrate and as a signaling molecule involved in plasticity and memory in mood disorders and in mood, let's say, and I didn't really show you this, but also neuroprotection. And so this is a kind of a summary of the mechanism. Some neuronal signals will trigger either glycogenolysis or glucose uptake, all lead through lactate production through aerobic glycolysis. Then lactate can be released by these transporters, and then lactate can be taken up by neurons through MCT2, of course, be transformed into pyruvate, produce energy, or also produce NADH, and at least we know that this has effects on NMDA receptor signaling. So this is a review that we published now, well, almost 3 years ago now, that really summarizes a lot of this. And yeah, so I started a bit by chance. I didn't know much about the brain, and for some reasons I decided to look at the glycogenolytic effect of neurotransmitters when I was at Seoul. That brought me to astrocyte because glycogen is only in astrocytes, and then brought me to lactate, and then to plasticity and neuroenergetics. And actually it took more than 35 years, but it's you know, a long way, but very exciting still now. Thank you.
Thank you very much, Pierre, for this really inspiring talk and for taking us into your journey, which is very interesting. So we can ask for some questions from the audience. Yes.
Just how long after learning do you find this changes?
What?
Yeah, how long after
Learning, yeah.
Yes, you find this change after change.
You mean in dendritic spines, for example, density?
Yeah.
24 hours already. But I tell you, we are looking.
No, because I was thinking about your mentioning consolidation.
Yeah. So short-term memory is not affected, but long-term, so 24 hours, maybe a bit shorter, but what we're looking into now and is that, you know, if this idea that learning, and there must be some kind of signaling, triggers this increase in the dendritic spine and increase in the energy facing them in the astrocyte, maybe to maintain a memory, you need to keep this arrangement for a long time. So now we are looking at glycogen granules after 3 days, 5 days, 2 weeks, 3 weeks to see whether it's kind of a permanent or long-lasting modification of glycogen.
You don't find changes before this?
No.
Question? I have.
Paola, that's it.
So you know that during the last years, let's say in the last 5 years, a number of papers came out regarding the heterogeneity of astrocytes.
The?
Heterogeneity.
Yes,
It says
yes,
that
yes.
astrocytes are not always
Yeah,
the
of
same.
course.
They have a little
Yeah.
So what do you think? I mean, if you have, do you have any comment on this regarding the metabolism? Is it depending on the area? Is every time the same or?
Yeah, no, it's a good point. I'm sure, obviously, there is heterogeneity. We looked essentially at 3 areas. Cortex in general, particularly prefrontal, frontal cortex, hippocampus, dorsal and ventral, and amygdala. So in all these areas, we found essentially the same. So it may be that it's specific to these areas, which I don't think, I think it's a very generic mechanism. Maybe different in the cerebellum because glia there is a little bit different. Maybe in the retina also because, yeah. So, but we haven't done an extensive survey.
Davide, may I another one?
Sure.
This is just a curiosity. You mentioned regarding the pathology, you mentioned a pathology where we have less lactate. Do you have in mind some pathology where we have too much lactate, in the sense that this shuttle is too, let's say, active, and then it's associated with something?
So I think there are you know, reports, particularly in Huntington's disease, for example, of increased lactate in the basal ganglia. What I think there, rather than too much lactate being produced, I think not enough lactate being used. And because you see in mitochondrial dysfunctions, you cannot use lactate. So if you still have some kind of signaling that will boost lactate formation, but you don't use enough, you will have more lactate.
So I have another question which is connected to this one, so I will give it now. I'm coming from slice physiology, and you showed that lactate is increasing the amplitude of NMDA receptor currents. So is it possible to think that actually the signal is the absence of lactate and not the application of lactate? So the signal is its absence, which will reduce NMDA receptors.
Good point. My view currently is that to optimize an NMDA receptor activity, you need many molecules. Of course, you need glutamate, you need glycine, you need to remove magnesium, and the next one, the almost next level, would be to have lactate facilitate its interaction with this campaignase. So it would be really a multi-factorial involvement to optimize an NMDA function. Now, if you don't have lactate, you see, it could be it's suboptimal according at least to our data. Yeah. So but maybe physiologically, we could see it is there because whenever you have glutamate release, okay, it will act on the receptor, but it will also be taken up by astrocyte and produce lactate. So you have a really kind of a 2-key system.
Yeah, in a way, we could add lactate to the Ringer solution.
Yes.
Okay, that's a good point. Okay. So I have another, and then I will give it.
But except that there is a problem. in many electrophysiological studies in vitro, there is 20 to 25 millimolar glucose, which is really, it's a very diabetic slice because it's 5 millimolar. No? And by the way, we're just doing now a series of experiments with Laurent Venance in Paris, because we started chatting after a talk and so on, and he was interested in spike time dependent plasticity and whether lactate would be involved, having seen what. And initially we didn't see much, and then I remember that. I mean, in the discussion came up this concentration of extracellular glucose. So he started again the experiments in 5 millimolar glucose, and suddenly, depending on the type of plasticity that he induces with this spike time dependent plasticity or just regular LTP, essentially the message is with strong synaptic load, you need lactate. With not too strong, you don't need lactate in 5 millimolar glucose. But then you can only rebuild it in the presence of physiological concentration, because most likely when you have 25 millimolar glucose, you just produce a lot of lactate endogenous. So that would be the, so it's not only add lactate, but also decrease glucose.
And I have another which is connected to this one. There is a long story about chloride equilibrium. Chloride equilibrium in some way is connected to the energy substrates. So do you think that this pathway can affect the chloride equilibrium, let's say, during development or during seizures?
Yeah. That's a good, I mean, because it will determine it, because the equilibrium potential of chloride is very close to the resting membrane potential, so it can be, as you know well, and all I can, so that affects GABA, essentially GABA signaling. So what we saw is that GABA does not trigger this type of coupling. So we looked at whether GABAergic stimulation of astrocyte would also result in the ANLS. And it's interesting because we looked quite deeply into this. What we found is essentially that the kinetics of sodium disruption, sodium homeostasis disruption, is much smaller with GABA when GABA is taken up by astrocytes. There is less sodium. It's much more transient. So the disruption of the sodium homeostasis in astrocyte is much smaller than with glutamate. And also, this is work by others, there is a significant reuptake of GABA by neurons. So it's more distributed, it's about 50-50 at least. Whereas for glutamate, about 80% is astrocyte.
Thank you. We have a question from the chat. I'll try to read because it's rather long. It's from Claudio Babiloni. Great lecture, changing the mind about the neuron-glia teamwork in cognitive process. Glutamate neurotransmission triggers astrocyte neuron lactate transfer. Memantine moderates glutamate transmission mediated by an NMDA receptor in the case of Alzheimer's disease dementia. Can you comment on the implication of your work on it?
That's a good point. That's a good point. Interesting. I really cannot answer. We have no idea, but it's a good suggestion. We can definitely try and see whether memantine has some lactate-like effects on an NMDA receptor on the NR2B subunits. Yeah, thank you for the question.
Any other question?
There is another one.
Yeah, yes. I mean, this is work by I mentioned in passing on oligodendrocyte, but also on Schwann cells. That is work by Klaus Nabel's lab.
Any other questions from the audience? Oh, Antonino, please.
Thank you for a very nice journey. Just a quick question. When you give lactate systemically, peripherally, how well does it cross the blood-brain barrier? Do you have direct evidence for that? And
yeah, good point. So it crosses. There are MCT transporters in the endothelial cells, but essentially it's about 5 to 7 times lower transport capacity than glucose. So hence the idea that lactate is good for neurons, but it cannot really access well from the periphery. You have to form it locally. The evidence for this injection of lactate IP, at least we also did it IV, but it's a bit more, cumbersome, is you use biosensors that allow to measure electrochemically lactate appearance in the hippocampus or prefrontal cortex. So indeed, when you inject the periphery, we monitor that there is an increase in the brain. Yeah. Thanks for
depression by introducing lactate.
in,
Can you use the lactate
good. Well, of course, we have thought of this, and in fact, just last week we had started before COVID a conversation with our colleague in psychiatry, and because we said, yeah, maybe we could, exactly what you say. The problem is, you cannot do IP injections in humans. You also, even in chronic administration, IV wouldn't work. So there are oral lactate preparations that we may try. We're just designing now a pilot study. We're not too confident that it will work, but maybe. But just to your point, once we were at Society for Neuroscience with the front of our poster on this with Jean-Luc and Anthony, and a Canadian psychiatrist came and said, "Oh, yeah, but this is well known that lactate is antidepressant." I said, "Oh, we haven't found anything in the literature except exercise and indirectly." And then he sent us a paper, 1947, from a really obscure journal like Rhode Island Journal of Psychiatry or something like that, and by two psychiatrists who did a study, but it's actually not even a study, it was an anecdotal report, N of 1 or 2, saying that they gave oral lactate, a mixture of lactic acid and lactate in milk to a couple of depressed patients, and they had improvement of the mood and sleep. And then, okay, the antidepressants came and nobody spoke ever about lactate. But the rationale why they did this trial, not really a trial, whatever, anecdotal test, was that at the time, electroconvulsive shock was widely used for major depression. And it was not done as it is now. It's still done now in certain places, and it works in very therapy-resistant depression. And they thought, and it was done without curarization, and so people were having muscle spasm. It was apparently quite horrible. But that they thought, okay, doing this muscular superactivity will generate a lot of lactate, and maybe that's why it is antidepressant. It's an anecdote.
We have a question from our Savior.
Yeah, you say that.
So thank you so much for your beautiful talk. And I have really two questions.
Sure.
One is more related to the last one, and then if it's maybe plausible to think about ketogenic diet to increase brain lactate. And the other one is more technical. So do you think that the lactate that you can add in an extracellular solution or in the brain, for instance, is going only on extrasynaptic NMDA receptor, or is going on synaptic into the synaptic cluster?
Very good question. Yeah. So the first one is actually, we're thinking a little bit in this regard, because what the ketogenic diet does, it will increase the expression of monocarboxylate transporters on the blood-brain barrier, MCT1. Yeah, A1. And because, by the way, ketone bodies use MCT, monocarboxylate transporters. They are monocarboxylate. So it's quite possible. I think it's a very good suggestion. Again, this is something I mean, there are reports of the efficiency of ketogenic diet.
Ketogenic diet is used for a lot of
improving cognition, improving. Yeah, absolutely. And I mean, people report better mood, more active cognition, and so on. So that's a very good point. And there could be a molecular basis. It's this increasing expression of MCTs on the blood-brain barrier. The second point, we're looking at this with Monica Di Luca in Milano, and she has this affinity assay she can see whether the NMDA receptors get closer to the postsynaptic density. It's an in vitro assay. Actually, it's done also by Fabrizio Gardoni, exactly, Fabrizio Gardoni. And we have had one very exciting experiment, or one set, maybe N of 3, 5, I don't know, that shows that lactate will increase this association between NR2B and extrasynaptic into close to the postsynaptic density. Then COVID came, and then the postdoc who was doing this left, and so now a new guy came. But they're doing the experiment. So if we can confirm this will be exact. I mean, that's what Monica, when I was talking to her, she got excited and said, "Oh, but maybe that's what is happening." So that's why we do these experiments, and they're encouraging. But it's just one set of experiments, and 18 months later, she's doing those experiments these days. Yeah. Yeah.
Another question from the chat: if the lactate supply can be effective in diabetic patients?
I don't know. Maybe. No, it's a good question. I mean, there is an interesting story around this, which I didn't have time to present. But what we found is that in Astrocyte, I mean, a glycolysis, there is a byproduct of glycolysis. So pyruvate is the main one, and then lactate. But there is about, I think, 5 to 10% max, is the formation of methyl glyoxal. Methyl glyoxal is a toxic compound, actually, that causes the glycation end products, glycated and glycation of proteins. And some people think that formation of methyl glyoxal is involved in the damaging effect of diabetes, because there is much more glucose and much more glycolysis and methyl glyoxal form. So we looked at this from other reasons, not because of diabetes. And what we found is that astrocytes have a very active glyoxylase system. There's glyoxylase 1 and 2, which actually eventually metabolize methyl glyoxal into, believe it or not, D-lactate, which is kind of interesting. But it's a minor component of glycolysis. Whereas neurons, they have a very weak glyoxylase system. So this is probably also one of the reasons why neurons cannot afford glycolysis, because if they form methyl glyoxal, they cannot break. So we have a couple of papers on this. And this is my only connection indirect with diabetes, because apparently methyl glyoxal is, some papers show that it's related possibly to peripheral neuropathy, diabetes. Yes?
It is known that the lactic signaling is affected during the obesity or binge eating or aberrant eating patterns.
Obesity and?
Or binge eating or
binge, yeah. Eating behaviors. Aberrant. Yeah. No, I don't.
Okay, so if there's any other question, we can thank again.
Really great to be live again. Really, it's fantastic. Ci vediamo più tardi, alle 3, per lei come andare a scuola, il primo giorno di scuola. Domani ci sono. Ciao, ciao, ciao.
La registrazione che esce la zona e basta. Sì, sì, come no?
Grazie a tutti per averci seguito. Arrivederci a tutti.